Well, good morning, everybody. So, for me, ASREML has been a bit of a story, so I thought I'd tell you a bit of the story because of where I'm coming from. How do I work this? That didn't work. What do I press?
Can you put it in full screen?
I don't know how to.
Right, okay.
Oh, it works?
Okay, Control L.
Control L.
Maybe?
Just the next slide, okay, it will move.
Anyway.
Did it work just then?
Well, I've gone forward. Anyway, so a bit of my story. I thought there should be a bit of science, so I've covered a little bit of the theory behind ASREML and variance estimation, and as a bit of plagiarism on Wombat, I've started another program which is essentially a rewrite of ASREML called Echidna. I think an echidna is a much more cute animal in Australia than a wombat. Okay, so is that going to do something?
Sorry, I'm not sure
What?
If you just,
Right. Well, I'll just keep talking and I don't know how you've managed that. Should be straight, simple PDF.
The Windows computer, nothing is.
Try F11.
Yes, F11.
oh, there you go. But I don't know if that will, does that work? Do you want to just try clicking the next arrow?
Right. Okay.
Thank you.
So I grew up on a farm in central New South Wales, very dry, long way away from anywhere, looking after sheep and driving tractors and so forth. Went to a small school. There were about 40 children in the school. One of my classmates, who was a few years ahead of me, ended up as the Deputy Prime Minister of Australia, but I didn't have much to do with him. And his sister I didn't like very much at all either. But then we had to go to high school, and so I went to Yanko Agricultural High School. That was a boarding school about 50 miles away on the river, established on the property of Sir Samuel Mackay, who was one of those Englishmen who came to Australia and took over large tracts of country and built a fantastic mansion. From there, not knowing anything else but farming, I got an offer of a traineeship at Port Kembla to do metallurgy, but then I got rescued from that by getting a Commonwealth scholarship and went and did agriculture, and spent the next 4 years doing agriculture at Sydney University. In the second year I got a traineeship with the Department of Agriculture, which at that time the state government was investing in agriculture instead of everything else that they invest in today. And so that led me to a career of nearly 42 years on their payroll, 3 years as a trainee, and then nearly 40 years as a biometrician, including 3 years doing a PhD at Massey University. And my job was as a biometrician. My responsibility was to help other scientists, whatever science areas that the department was into, whether it was weeds, animals, plants, all kinds of agricultural experiments, to analyse those data. And so that collaboration gave me, over the years, 120 publications. The vast majority of those are applied statistical, my name's down the bottom because I did a bit of statistics. But to do those statistics, I needed software. And this was in the area where software wasn't everywhere available. At one stage I was using a dial-up system to some computer in America. And when I moved out to Trangie, to get onto the host, I had to ring up a manual telephone exchange to get a dial-up link back to the computer. And that was a bit awkward for the telephonist, who every 3 minutes would say, "Are you extending?" And of course I wasn't listening, she'd just get a scream in her ear. But anyway, so I took on a role of writing software which the group used, but which I could use. And there were lots of programs written. One that lasted probably 20 years was REG, which basically did regression analysis. Other colleagues wrote an analysis of variance program. And when breeding, after I finished my PhD, the adoption of breeding in the lamb industry, I wrote a little program that could be taken out onto the farms so that a scanner could go out, weigh some lambs, take a fat depth measurement with an ultrasound, type in the data and give the farmer a report. But that program quickly became used to give comparisons over multiple years and provided a basis for a complete transition of genetics in the lamb industry. And the same program was used for the wool industry for wool selection over from 92 to 2006, before being taken over by AgBoo. Now, while I was doing my PhD, the software then, that software then led on to ASREML, and I've just summarised it there as a program to fit the generalised linear mixed models to research data, all the kinds of data that I was being confronted with. So I wasn't just thinking of animals or plants, but whatever came my way, insects, whatever. But it covers spatial analysis of field experiments, because that was an area that was being developed at that stage. We wanted to do multivariate analysis, not just univariate analysis. The background of this was a multi-environment trial analysis, animal models, and then we have the genomic models further down. So ASREML was developed with a lot of support, and I'll talk more about that. But where is it going to? Initially, I developed it while I was employed by New South Wales Agriculture. Since I left the Department of Agriculture 9 years ago, I've been continuing to support ASREML, initially unfunded, then funded by the University of Wollongong, and for the last 4 years funded by VSN. But I found this rather frustrating because, I sense a conflict between my purpose of writing ASREML in the first place and the commercial reality of it now belonging to VSN, because they purchased the product just after I left the department. And so I've started writing a new version which I'm calling Echidna, and VSN have recently decided that it is in their interest to cooperate with me, and the idea will be that Echidna should feed into the continuing development of ASREML, but also provide me with the opportunity of doing things and providing software for people to use for non-commercial purposes outside of the framework of ASREML. So hopefully that will be available within 6 months. I've been working on it for 12 months so far and made reasonable progress, but it's not really complete yet. The issue, the philosophy that I've had, though, I've summarised in those words that I've had to think about. And really what I'm here for is to do good to as many people as I can. While there is an issue of people need to get paid, and I like to get paid as well, yet also information that we receive we need to pass on to others. And we really need to believe in what we're doing. So what is not of faith is sin. So that's been part of my journey for the last 6 months, thinking about how one relates with VSN. Okay, so a little bit more about Robin. I put this in a presentation once, and Robin did not like this picture at all and promptly gave me another one which he did approve of. He thinks I'm winding him up.
It's the first time, when I saw it, it was the first time I saw it, but it's a good summary of me and you. Here you are trying to wind me up again.
So we've had an interesting career. I first met Robin, I believe, in 1981 when I was doing my PhD, and I think he came to the World Sheep Congress that was held in New Zealand. I don't particularly remember much about him, but at that point, except that I did negotiate to go and visit him in Edinburgh here in 1982, where he showed me, well, I stayed with him at Roseneath, and he looked after me, and we talked about my PhD work. Then he kept dropping in on me. We spent 2 weeks together at a Gianola workshop in 1984. I don't quite understand why Robin was the first person to in our guest when we moved to Orange as a house guest, but he was with Harriet, and Robin just kept coming back and coming back and coming back. He doesn't come back to me so often these days, but I come over to see him. And so this was at Orange, and I've spent time with him at Leith, spent time with him in Harpenden and at Redbourne. So the ASREML began when Brian, who you'll hear from, was trying to analyse 16,585 mean variety yields that he extracted out of yearbooks that the department had produced, summarising variety trials that had been done. There were 1,077 trials representing 60 locations. There were 110 years of data, 107 varieties, and it wouldn't fit in GenStat. And so I said, oh, I know how to do that. We can use derivative-free stuff that I'd kind of learnt from Robin and Karen Meyer. And so I wrote a program that could analyse the 7 variance components that were involved. Well, Robin dropped in and said, oh, we can do better than that. We can do average information. And so he told me what to do, and I changed the program, and within a day we had some average information results that were quite satisfactory. So he went away. He went and told this idea to people in New Zealand, and went and visited Brian and did some various other things. But next time he visited, I had adapted or rewritten reg to now utilise the average information in a program I called REML. But there was also another program called REML in New Zealand at that stage. And as we know, Robin had also shared these ideas, and he later told me that he thought the Finns were going to develop the major software that would be used. But anyway, I didn't ask him, I just did it. And so the idea is a bit awkward with not having that in it. It is the average information algorithm is that we want a second derivative based method. The two common forms and traditional forms are called Fisher scoring and Newton-Raphson, and these both have trace terms of four-way products which are very hard to calculate. It'd be expensive to calculate. But if you average those two algorithms, the matrix, that nasty term cancels out, and the result is that you have something that is much simpler to calculate. So in simple traditional models, it exactly drops out. For more complicated models where you do have products of terms, then we ignore the high order terms. And so I'm hoping to write I've drafted a paper which is going to spell out some of the things that I just mentioned, in more detail, some of the things that I mentioned here. Now in '93 I came to Roslyn. That's when I spent a month or so living at Leith and catching the bus out to Roslyn, and then moved into the village of Roslyn for the next 5 months, and I'm working on a program, MTRAM, to include carving ease statistic. And during that time, Brian visited and we closeted away for a couple of days to write the 1995 biometrics paper. Now, but Brian at that stage was then my boss in the Department of Agriculture. He was head of statistics or supervisor of biometry or something. And as the group, we had a continuing need to fit models in all of our applications. And so the idea was developed in discussion with Robin on his visits to extend ASREML to handle direct product structures. And the common ones are a variance matrix times identity for trait dot genotype. We can have multivariate residuals, so that leads to a multivariate analysis. We can have a multivariate animal model type analysis where we've got a relationship matrix. And the key area was also the inclusion of spatial analysis where we have a correlation structure on rows and columns in the field. And so I took the shell of ASREML and rewrote the core of it, and that's how we managed to create ASREML in effectively 8 months. So my motivation though was always to analyse the data that I was being confronted with on a continual basis. And so over the next 20 years we've had a team where Brian and Robin and I would get together on a fairly regular basis. I was writing the code, but Brian was very much involved in presenting challenges to models that needed to be fitted and getting other people to write up the theory of what we were putting into the program. And Robin always had new ideas to contribute because the foundation of all of it was what Robin had done. And of course, I haven't done everything that he wanted because I can't keep up with understanding everything that he wants, which makes him cross from time to time. I'm sorry about that, Robin, but we do what we can. So, none of us would have happened unless Robin had kept coming back to the centre of New South Wales. And I was going to tell you a story here, but I'll save it for tonight. But he's taken an interest. He's been extremely patient over 36 years. But there's also been other people, and I just mentioned Brian, who prompted me to go and do the PhD in the first place, so he got the whole thing going, and included me when I visited in '93 to go and do some work at Genus. And Karen Meyer, who I've never actually worked with, but I've plagiarised greatly. So the underlying algebra, for those who are not familiar with it, is that we've got a linear model and we've got a variance structure for the residuals, which can be linearised as a residual matrix plus a product of a design matrix for the random effects. And we define a likelihood function for that for the normal distribution, which is given by that expression. And if we plug the data in to that expression, then we've got some variables, the parameters in that model, and the parameters are the fixed effects Tor and whatever variance parameters have gone into the definition of V, which are R and G. Now, so the process is we can estimate the fixed effects and calculate estimates of the random effects. Now, assuming that we've got a set of parameters for the variance parameters to start with. So we need to start with some initial values. And then we can use the residual maximum likelihood. Am I causing that to jump up and down?
No, sorry, that's me.
Which means maximising an adjusted likelihood which takes into account the loss of information from the fixed effects. And so the log likelihood that we then have is given by that expression, and then we need to differentiate that, and I won't go through the details beyond that, but we use the mixed model equations to do the process. We have some initial values, we solve for the fixed effects, we evaluate the likelihood, we get some updates for the parameters, and go around the circle until it's converged. Now, the first kind of method then that I was using was derivative-free algorithm. The E algorithm is also used, which uses first derivatives, and Robin's original paper was based on a Fisher scoring approach, but the average information algorithm is much simpler calculation. It still has the properties of quadratic convergence, and so has proved a very successful method. Now, so ASREML was designed as something that would cover a very wide range of analyses, from simple regression, analysis of variance, multivariate analysis, prediction of genetic effects, and the power lies in the broad range of models that can be set and the variance structures that can be applied to it. So I'll give more details of some of the core of that in the written paper, but just to introduce some of those subjects, the next few slides. The actual algorithm that is based on is that you form the sums of squares of products matrix for the mixed model equations. You add the G inverse matrix. A key part of ASREML is to maximise the speed, which has been involving using sparsity, and so you need to determine an efficient order for solving the equations in a process I call absorption, which then lets you calculate the solutions and the residuals, from which you can calculate some working variables and their cross products, and so you can absorb those cross products into the AI matrix. And then you can complete the inversion of C, so you can calculate the score, and then you can update your parameters. As Es has already mentioned, a key issue is the parameter space, because the algorithm really doesn't know about parameter space, and often the maximum of the likelihood can be somewhere where you don't want it to go. Now, the analogy of that is that if you do a fixed analysis of variance of that, and you've got blocks and residual, it's not unusual for your F statistic for blocks to be less than 1 if there's no block effects. Then half the time, because the block variance is just an estimate of residual, or if it's small but it could still have a negative an F value less than 1. But when you convert that into variance components, that means a negative variance component. And often we like to keep our variance parameters inside a parameter space. So what ASREML does is when we get some updates, we test the parameters whether they're still in the parameter space, and if they're not in the parameter space, then we can either restrain all the parameters or alternatively pull back the extreme parameters and condition the other updates on the modified updates for those that went out of bounds. Okay, so the key part of sparsity is that we don't need all the elements of c to the minus 1. And so, speed up. So what we only need is those elements of c to the minus 1 which actually exist in the C matrix before we invert it. And so that was a major save and of memory as well. Factor analytic models have been very important also in the parameter space thing, because the factor analytic model gives a variance matrix that can be guaranteed positive semi-definite. And then we've had an explosion of marker data in recent years, and this is where ASREML has really not kept up. So we can calculate from a marker matrix of 0, 1, 2s, a G matrix as a cross product of the markers, but this G matrix is dense, and ASREML will spend a lot of time working with sparse matrices, so that's a key area of the focus of the development of Echidna, which implementation would feed back into ASREML. So why Echidna? Well, needs to get, the code of ASREML is 20 years old. It's time, I believe, to get rewritten and sorted out, and to accommodate the models of the next 20 years, so that more people can access the technology to explore this incredible world and to improve agricultural productivity. So the idea will be that Echidna will be free for non-commercial use and will feed into the future development of ASREML. And I hope that something that's reasonably covers a broad range of what ASREML will do will be available by the end of the year. So you can get it and play with it at the moment. The problem with our implementation is that some email addresses seem to get blocked, so that if you go to that website and try and download it and you can't register, try a different email address. So that's what an Echidna looks like. And I'm continue to be supported by VSN to support ASREML and permitted to develop Echidna. Thanks, Lisa.